if I could remind everyone, this meeting will be recorded, so if you don't want your image included in the recording, please turn off your camera. As well, just any questions that could be put through the chat while the talk is going on, and we'll go through questions after the talk has finished. Okay, right, it's my very great pleasure to welcome Viktor Korzun to give the talk on application of genetic and genomic research in cereals breeding. Viktor has more than 30 years experience in the development and application of molecular markers and novel breeding technologies in cereal crops. He has a PhD in plant genetics from the Institute of Genetics and Cytology from Belarus, and he was a visiting scientist at the Genomics and a postdoc researcher at IPK before he moved to KWS in 1999 to head up the cereals breeding technology department. As of 2018, his current role is as a global lead scientific affairs with focus on national and international collaboration. Viktor, the floor is yours.
Thank you, Luke. Thank you, Robbie, for this very kind invitation. Now I give it my slides, and this is not the first one. We need to move a bit. 1 second, please. Oops. Now you see this start slide. Look, just show your fingers. This is everything is working. Perfect. Good afternoon. This is my great pleasure to see so many friends, so many breeders, and so many young scientists here. I hope I could make it in my talk today a little bit you more enthusiastic on plant breeding and also on specifically on application of genetics and genomic research in cereals breeding. This is a great pleasure to talk. This is on International Barley Hub, and Luke already made my introduction, but I started first from 3 crops you see on picture here, and looking where's the pointer. There's a pointer. This is my famous crop, rye, in the middle of wheat and 2 barley types, 6-row barley and 2-row barley. As Luke already mentioned, I started my PhD in National Academy of Science in Belarus. Then I was invited to Gatersleben, as most of you know, as flagship of plant research, and also a lot of cooperation with Dundee specifically. I was so lucky to use every year the possibility from 1992 till last year to visit John Innes Center. I have really privileged to work with Dave Laurie, with Miguel, Catherine DeVos, and many. And currently, it's not secret, I'm on scientific advisory board of John Innes Center, which is make me really proud. And as already Luke mentioned, I was for closely to 20 years head of breeding technology in cereals, meaning to work on all crops, what you see here on the picture, and from 2019 moving on functional global lead on scientific affair. In the end, if somebody interested to know what is behind of this function, I'm happy to explain. By the way, this picture I took specifically because variety Julius is one of the variety winter wheat variety which was sequenced fully with pangenome of wheat. Few words about KWS. And we are very clearly seed specialist, working on sugar beet, corn, cereals, and other field crops, I show you later, and vegetables crops. KWS think in generation. That's the reason I like really underline. Currently, we have our speaker of the board who is from 7th generation of company owner. And this is give us possibility to think in long-term goals and take it very seriously, sustainability success. I allowed to show you very short film, and I hope because you will enjoy that.
Hello, may I introduce myself? I am the seed, strong, healthy, and powerful. I bring life and have prevailed among countless varieties. But to become accomplished, I must travel a unique journey. My journey is long and takes many steps, behind which are clever and patient minds. Scientists who question, research, and develop, repeating their methods over many years with foresight. Breeders from KWS who always create new and improved varieties, like me. Thank you for being so committed. Thank you for your patience and passion. You anticipate the future, while at the same time, you draw upon your great treasure of experience. You act out of tradition and are open to all facets of my nature. Without you, I could never set out on my long journey, over land or over water, to arrive where I am needed, in farmers' fields. You are in close contact with them, sharing your knowledge respectfully at eye level. Your actions allow me to achieve my greatest goal, to fully develop on-site under increasing challenges. Adapted to climatic changes and robust against diseases. Together, we then let something very special emerge. Valuable and sustainable food for current and future generations. And what makes me most confident? There are many more of me growing in the fields, high-yielding and in many varieties. Together, we feed millions of people. We are the seeds, and we bring life. Yesterday, today, and in the future.
We are the seeds together with you, and I love this film, and I know from many students now they love also, and this is important for us also to communicate with politicians with other level of dissemination. Back to the story of KWS, we have story more than 167 years now, started from sugar beet in small village called Klein-Swansleben, and that's the reason why company name was in beginning Klein-Swansleben Saatzucht Rabetken Wieseke, which is impossible to use for international. That's the reason why mainly we use now 3 letters KWS. Them cereals, which have a different story with Ferdinand von Lohoff in small village in Petkus, who is one of the king of the rye breeding, moving them after in the end of Second World to Einbeck from east part Germany. From east part Germany to west part of Germany. 95, very smart and very important decision to start with corn, and we should mention now we are very successful with this crop as well. And 2019, another step forward to start with vegetables crops, which is I could explain later on. Today we are on 70 countries worldwide. We are from last fiscal year, and this is our philosophy or show last year before. We have 1.5 billion. We are in more than 70 countries, more than 5,000 employee, full-time employee, and very important for you to take this number about roughly to 80% reinvestment in breeding and research. And I like underline, if I'm talking today about research and development, I'm talking about breeding and research supported plant breeding. We are currently on position 5 worldwide and estimation of position 3 in Europe. We are working on 11 from 13 most important crops. You will see here in orange global acreage and grau is global production. And you see this is corn from production is number 1, but from area of cultivation wheat is still number 1. We are not working on rice because it's in different climatic condition, and we are not working on sugarcane, which is main competition, and we not believe on sustainability of sugarcane in main competition of sugar beet. We put it vegetables in as one crop, but we are started from scratch on 5 most important, and we have some other small vegetables crop by acquisition. I would talk today about wheat, barley, and rye. As I already mentioned, we are seed specialists, and for that, for us, it's extremely important to have innovation. We're talking about sustainability. I already show you independence and profitability. By the way, I change the order. If our commercial people talking, they're talking profitability first and then innovation. For me and for us, extremely important, and plant breeding is innovation. Because having EU farm to fork strategy set target by 2030, and you know as good as me, this is expected to have by 2030 reduction of chemical pesticide by 50%, reduce of application fertilizer by 20%, achievement of 25% of organic farming. But what this mean for the European agriculture? And we try to project that, and this is what mean if this full implementation of farm to fork strategy, production losses in Europe will be around 23%. We will reduce also export and lower competitiveness of EU farmers. In global scale, agriculture prices will be increased, which is led to decline food security. And to compensate that, and this is not our numbers, this number came from a public domain from very famous FFFA research. You see citation here. We need 2 decades of plant breeding to compensate that. This would mean for us, we need and we should make plant breeding to be more efficient and more faster. Now I move into another part of my talk, a few words about global trend, because farm to fork is our European strategy. We should and we have responsibility for food security on global level. I would give you a few examples of current breeding targets by using wheat breeding program to make for people who spend a lot of time working on barley or another crops attractive. And I would talk about new breeding strategy and supporting tools, which is related to all crop I'm talking. Now a few words on global scale, which I think I don't need to repeat for you again. We expected the population growth, mainly coming from not European country. We also see the shrinking of per capita average, large city like London, Berlin, they're growing, but they have less agriculture. We see the climatic changes. Also, this year is completely different to last year. We have so mild winter, but now very cold spring. We also know that about 52% of our yield, which is already on the field, we're losing due to insect, fungal disease and storage damage. And as already mentioned before, we have limited resources, water, fertilizers, and plant protection. And again, to remind you about Green Deal, farm to fork, 20% less fertilizers, 50% less plant protection, and this is everything by 2030, which is already in 7 years. Talking about wheat breeding program. Started from beginning to mention specifically for young scientists, 2 major crucial points in plant breeding and in cereals breeding. This is the variation. Sometimes I mention if we don't have variation anymore, we may stop our breeding program. We could increase variation by taking blood genetics resources, and we hope that we may increase some targeted variation by using new breeding techniques like gene editing. Another step is selection, and this is depending on the country, depending on the size of your program. This will take years, place, and replication. After that, in many European countries, is going for official testing. Finally, is variety, which is depending on the crop, 8 to 12 variety. It's a really long time. And core formula of plant breeding, this is the genetic gain. This is what we expected, the advantage every year from genetics, from other genetic related topics. And this is formula related with L, which is generation interval, and I would repeat L a few times during my talk: selection intensivity, accuracy, and genetic standard deviation. Selection intensivity. This is quite easy sometimes to think about. If you make your program double bigger, you may be more successful, but bigger meaning is more costly. Accuracy, very important. This is the reason why we again came back to high throughput phenotyping by using different type. And I will show you in the end what we do in this direction. Variation, I already mentioned about how important variation, but to have too much variation in target program. And I think experienced breeders like Heidi with us today would completely support this. Is not advantage, it could be disadvantage. And again, time have linear effect. Talking about wheat, and usually during my presentation, I mention yield, quality, resistance, agronomic traits, meaning yield stability, and now became also in Europe specifically very important nitrogen use efficiency. And usually I'm asked to make image to close the eye and think about iceberg. And this is everything what I show, the upper part of iceberg, because looking down underwater, you will find a lot of factor here, which is related with yield and yield stability, disease resistance. For example, I pointed here for wheat most important in Germany, for example, and talking about quality, we have different parameters of quality. I think the similar projection we could do for barley, specifically for malting barley, because for me, after 30 years experience, still the open question, what the step of selection should be done first, quality, talking about malting barley, or yield, or how to combine is the best knowledge-based way to keep the variation from one side, from other side to be on top of quality and top of yield. Moving to wheat breeding, I show you specifically for the people who not involved in wheat breeding or cereals breeding, a couple of steps of practical plant breeding, in this case wheat, but barley, as I already mentioned, could be similar. You see young people who is very happy, but doing very difficult and not easy job, because you need to take answers in each spike 3 to be able to pollinate, to make F1 generation, to make variation, what I mentioned before. And then, extremely important to develop homozygote material, because to make it homozygote is easy to select on different level. And you see here a presentation of, it could be single seeds the same, but it's specifically very interesting to have double haploids. And then million of plots. And in this stage, I always make the difference, show the difference between farmers and breeder. A breeder always happy if we have difference. For example, you see here rust, you see here plant height, because from one stage to another, from one side to the another, is aim to reduce 90% of material to keep 10%, which is most suitable for target region. We like finally to provide the variety. And finally, the combine measure everything. And for 20 years ago, I have very funny discussion with our famous breeder on cereals who mentioned, I'm not care, the 5% yield came from photosynthesis or from root system, because combined will measure everything. But finally, we was able to show him this is 2 independent QTLs. And if you combine both, you could get it not only 5, you could get it 7% or 8% of yield advantage. After that, we don't have any more discussion about, and we see already the possibility and potential of techniques by using molecular breeding. Probably interesting for you to know, here I put it in comparison. 1 hectare of field from left side and from right side. From left side is 1 hectare of farmer field, and usually, because in intensive farming I'm talking here about, is farmers use 8 hours per hectare. As breeder, we use 600 hours per hectare. It's time consuming. That's why we're interested on all new technology, which is make simplification, which make precision. Talking about wheat in Germany, and you see the graph started from 1980. I was not make updated here, but you see this, we still have increasing of yield. And what is interesting for you to see the most high yield, which was recorded in New Zealand 2020, was 70.4. Last year, this yield was beaten, and we are on wheat closer to 18. Because here is a lot of from barley society. I also put it one text here related to barley, which is 16.2 ton per hectare. Again, reason why I'm talking about the highest yield, because if we look on wheat yield around the world, we are above, below 2 ton per hectare. That means we still have a lot of research need be done in different regions of our planet to keep the yield stability and yield potential for a couple of other countries as well. But also for European country, you remember what I showed you regarding farm to fork. Talking about wheat adaptation, France, the largest country with winter wheat production, 5 million hectares, Germany followed with 3 million, UK about 2 million. And this is 3 program, our European program on wheat. We have 4th one, which is United States. You see the program is connected, but program is not overlapping. The reason why, because depending on the country, depending on the condition, for example, UK, we're looking for very short plant type, yellow frost sectora, white sowing window. Sometimes in December, we still could make the sowing in UK. In France, I always mention, if you don't have quality, we will not release the best yielding variety. I borrow one slide from my colleague Klaus Olda. Here is a similarity to barley, and this is probably one of only few slides I'm talking about barley. Again, we have a couple of programs here in Europe, which is related but not overlapping. For spring barley, situation could be different. And that's the reason why I'm personally always mention the spring crop could fly from one country to another. For winter crop, it's much difficult. But winter crop from other side bring more yield because extension of vegetation period. And for barley, I think we have a couple of years ago very clear data in Germany. This is at least 25 till 30% more yield advantage. Coming back to wheat and coming back to quality, I already mentioned for you is different type of quality. You see again UK here, you see the France, where the majority is BPS, and Germany, which is a group A, most requested group of wheat by farmers. Again, coming back and remind you about the original slide, again, the formula, magic formula of plant breeding. And that's the reason why we are so interested in new breeding strategies and supporting tools. And today I would not talk about the crossing selection. I would not talk about hybrid breeding. I give you a few slides about cell and tissue culture because it's extremely important. This was a really pioneering and excellent step in plant breeding, specifically by cereals breeding. I would talk with few slides about molecular breeding. I would not talk today about GMO, genetic engineering. But I would show you advantage of genomics and how we used in a couple of example genomics. And of course, I have few slides about new breeding technology, specifically on gene and genome editing. Cell culture is not any doubt. I think Heidi, you was one of the pioneer of together with your growth of using of microspore culture specifically for spring barley, but we are so happy on barley. This is a methodology which was developed by Professor Ken Kasha and Professor Lörz from Hamburg, is so excellently working on spring barley and winter barley. It's work now in our hand, at least on winter wheat, but we still, after many times to try, could not make the methodology using in routine on rye, because probably biology of rye. And for young people who is not aware about the technology, you see the technical assistant sitting here. Here is a spike. And from spike, we are taking microspore and from regeneration produce unlimited number of green plants, which is excellent because next step after is going for the selection. And what this mean, you see from left side on wheat, you have so-called pedigree-based scheme. And it's usually taken 8 years before for official testing by government. And having double haploids, we could reduce this in 2 years. Similar situation of as I show you on barley. This is really excellent. This is breakthrough for us and we are very happy to use the technology to combine now with genomic selection and a couple of other applications. selection was, is, and will be stay as very important, because I think we could not make selection in artificial condition only. We should go to the field. And if somebody asks me how you follow as breeders the changes of climatic condition, my answer is we are not making selection only in greenhouse, we make a selection on the field. That means we are follow climatic condition. But sometimes it's not so easy. If you look here, this is winter killing. Winter killing last, if I remember correctly, in Germany, we have 2012 or 2010. But in east of part, like in Poland, we could have such observation. And if we, for example, have most yielding variety of winter wheat, or which is still more difficult to have winter barley high yielding, but without frost or winter hardiness, we have problem in East Europe, specifically in Poland, for example. Another example here, yellow frost, because till 2014 on wheat was not the question of yellow frost, but changes of different races. Yellow frost became the problem in question how you select it. In some case, and I took this slide specifically because fusarium and toxin now on the level of regulation of EU, that is mean zero in baby food, and this is very difficult to select. Here we're using the artificial inoculation, and our strategy of the company, we are not releasing the best yielding variety if we have a high level of susceptibility for fusarium. Few slides about molecular breeding. I think in this cycle, I don't need to go very deeply about using of different part of plants, seeds or leaves, using of CASP technology for allele-specific marker. We could differentiate very well for disease which is must be, in this case, for example, virus, where the phenotypic selection in field is very difficult. I take time before preparation my lecture, and I took very famous Big Mac 29, Robbie, which was my first marker 30 years ago. And this was really beautiful. Thanks once again for your development. This is microsatellite. Some of people, young generation, don't know anymore what is microsatellite, because all technology based now on CASP, but this was a really good example. I should omit in this stage that 30 years ago, we, and I personally, was probably too optimistic, thinking that every marker which is 5 centimorgan FVF on traits could be useful. Now we are talking about CASP, which is allele specific, which is completely 100% make prediction. But we are very happy because technology is developed, and I show you how to use it. Another example related with barley specifically, and this is completely different virus. This is barley yellow dwarf virus, and I think barley breeder will confirm we have so less variation in our Hordeum vulgare pools. That means we need to look on Hordeum spontaneum and Hordeum, in this case, bulbosum. And we are very happy that Richard Pickering, our friend, made early stage of a different type of introgression. In one case, we found the gene which shows the full resistance. And what is mean in case of barley yellow dwarf virus? You have yield or you don't have yield at all. And this is very important. Again, showing as nice example, in some case, we need to go far from our pool to search on relatives, white relatives crops. And this was very and still some long way, but new technology, new possibility help us. Plant genetic resources was, is, and will be our gold sources. And here you see from left side as the field in EPK Gatersleben provided by Professor Andreas Börner, variation on wheat. But you see from other side, a slide provided by our breeder Erhard Ebmeier, and we still have variation. I'm not agree with people who mention you as elite breeder, you lose the variation. We still have variation. And you remember too much variation in your pool is not advantage, is disadvantage. But now we have really nice tool to monitor and manage of genetic diversity. And one is very good example, a recent publication of Joachim Reif group in Nature Genetics as based of a very famous in Germany project on wheat with name GeneBank 2.0 now. And this is project which is already going 8 years. And I'm personally very proud because in this case, we show for Ministry of Education and Research Germany, if we started with plant genetic resources, it is not possible for us and we could not promise in 3-4 years project to deliver. In this case, we agree on scale 3 + 3 + 3. That means after 3 years is evaluation. If it is scientifically and industry still this good result, is they have extension. And we are really proud that a couple of projects, one of them is GeneBank 2.0 or now GeneBank 3.0, and another one, SHAPE project on barley, which is Luke and Robbie, I think very well known, coordinated by Neil Stein. Genomic selection is not doubt. This is our top tools we are using, and I would not repeat probably only a few words for young scientists. This is a really strong and very powerful technology. We take it from animals breeding for a specific reason. We use phenotyping and genotyping and put it through the biometric prediction model. Most of them working very well, and then we could pre-select genotype. We may not go on the maximum of best of the best, and we should very careful here be with variation, because we should keep the amount of variation we need for success of our breeding program, not only for 2 years, but for 10 and more years. We started very early, and what I could tell you right now that we are applying very successfully genomic selection for all crop of TWS, and here is my example only on wheat, barley, and rye. Again, remember this is magic L, because genomic selection help us to have more accuracy, to have shorter generation interval. And here is very short selection of publication. I was involved with my colleague from EPK or other institution on yield, quality, and resistance. And the recent one, which just came now through Science Advance, and using unlocking big data for prediction of grain yield in hybrid wheat, which is important as well. What has happened? 30 years ago, we used BMAC 29 and a couple of other markers. Now we are talking about genomic selection. We have an unlimited marker. And this is possible because during last years, we have really a revolution and development of the tools. Here you see, and this is again not secret for anyone, this is two possibilities: use a CASP or array-based genomics selection, or for discovery larger chip. And what we have, no limitation on number and distribution of the marker, and dramatic cost reduction. Because anyway, we are on competition with plot. If I as breeder could make more accuracy and more efficiency selection on the plot, I will not go to molecular breeding. But we are now in lucky situation that this molecular breeding could support, and the cost for molecular breeding is really low. This was not possible without a really strong development on sequencing. And here you see the title of Nature, Science and Plant Journal for most important crops like barley, wheat, and rye. And in some cases, as on wheat on pangenome, you see we also was involved with one variety. And I'm specifically very proud on Plant Journal because this was the first sequence of rye genome, which is in comparison with barley is larger. If I remember correctly, Robbie, barley is 5.6, rye 7.8 GB. And that's the reason why I will show and share a few things from rye, Secale cereale. And I am always referring to first rye map, which was produced by Professor Günther Fricke. You see him in picture with his wife. By the way, they established Günther and Anna Fricke Stiftung, and young scientists and some already professors receive. I think Neil Stein was one of them. Please pay your attention on chromosome 2 only, because on chromosome 2 you see two isozyme marker. And I remind you, this is 35 years ago. It's not too long. Then 20 years ago, we published with Andreas Börner another map with some microsatellite chromosome 2, some some traits. Some of you know this trace. This is Ligulus, Z locus as well here. But then the group of scientists very enthusiastic. Four professors, Professor Geiger, Professor Chris Caroline Schoen, Professor Thomas Midaner, Carl Schmidt, but also what is very important, young scientists, produced, developed of first array and produced now map with 135,000. Again, chromosome 2, you can't see any numbers, you don't see any numbers, but question is what for? One very good example that the gene DW1, which is not one-to-one what is RHT1 and RHT2 because it's GA sensitive, RHT1 and RHT2 from Green Revolution is insensitive, but it's also now fashion and in modern plant breeding because we could not apply too much chemistry now for plant reduction. And this is really good because we published with Andreas Börner 96, again, more than 20 years ago. We don't have too much markers. We know it's chromosome 5 long arm. But after publishing of first SNP map with many markers, my colleagues in very short time found candidate gene for that. And this is really extremely, I know Luke and Robbie, you have many such examples on barley, and we have many examples on wheat and other crop, but this is a show how is a technology work in hand. Another point which is I already always like to show, this is pollen fertility restoration on hybrid rye specifically. And here, talking about ergot, this is in German language called Mutterkorn, and this is again poison, because it's very strong alkaloids. And this is alkaloids was collected in the early 60s by pharmacy and was only the possibility to prevent of a pregnancy. And you could imagine what could be happen if somebody buy the organic bread of rye, but with some amount of ergot, specifically for women who have the pregnancy. And this is not a way to have ergot in, and we'll try to reduce. But unfortunately, we don't have too much resistance for ergot. But what is good news, what we recognize in early days, this is biological competition. This is so-called escape, and breeders used in some cases, in many cases, escape because not care about biological mechanism, because it's difficult, complex, we don't know, but if it is work, it's really nice. The pollen fertility restoration came from a very exotic rye called Iran 9. You see here is like grass, and elite line is from right side. That means looking here two plants, again, please concentrate in this case on chromosome 4, because we're looking for this green piece. Rye have 7 chromosomes. Both plant A and B is in BC1, which is theoretically should be 75% of recurrent parents, which is elite line. But look, to check with markers, we may have found the plant number A have 87% and plant B is only 64. Both of them have our targeted region. But if we selected A, we already 1 year early. And this is again advantage for crop like maize, rye or hybrid barley to speed up backcrossing program. Talking again about ergot and what has happened, this is the first hybrids with a lot of ergot. Population variety was always golden standard, and you see this new pollen plus strategy. My opinion, this is one of the best examples by using plant genetic resources, molecular breeding, and knowledge of plant breeding to produce product, which is very asked by farmers. And I updated, put it where it's still not published, but we have some additional information. You will see here this orange makes the hybrids which is have RFP1, which is restoration gene, and hybrids which is doesn't have RFP gene. And this is the restoration index from one side, from other side ergot severity. And you could very easily see, depending on the year, of course, because some environmental condition, this is hybrid with RFP1, pollen plus have advantage for the farmers. Again, what has happened during these years? Genetics has become genomics. Sequencing of large genome became a reality. Bioinformatics, data integration, biometrics challenges are enormous, and precision phenotyping came again. That's the reason why I took again few slides about precision phenotyping. But before to talk about that, for me, specifically for young people, it's very important to put it this sentence. Translation of the genomics information into breeders' knowledge is essential, because we are going in more and more complexity, meaning we are talking not only about a plant, we are talking about soil, we are talking about a microbiome, and we're talking about environment. The complexity is increased, and we are definitely need to share in open way the knowledge. In our case, as I already mentioned, R&D support of breeding from beginning to the end. What it means is specifically for gene editing, because for gene editing it's not enough for us to have only a structural knowledge. We need to have the functional knowledge. And this is a slide not on barley or wheat, on sunflower, I like very much, demonstrated what could be the difference. In very simple words, you remember magic formula, again, L, we could short the time we need to have to less plant. And I think without such technology, we will not able to recover our amount of productivity and to be sustainable as expected by farm to fork. One very good example, and all German breeders involved on that, named Pilton project. If somebody interested, you could search in Google, you will find Pilton, because it's in German Society of Plant Breeding, it's very well posted, and aim of Pilton for wheat, to develop wheat by using genome editing for broad fungal resistance for 3 major diseases on. And I could tell you in this stage, we are on good way. I mentioned that digitalization and R&D trends, improved analyze large amount of data, water and soil data, satellite and drone technology, new tools, exponential development sequencing, better breeding decision by meaning improvement data integration and interpretation. What does this mean? We need to have digital phenotyping on different levels. Level 1 is for research, for example, plant physiology, where we use the phenotyping on single plant level in a control condition, then moving to plot level, which is meaning plant counter and breeding and research, and finally on field level for the farmers. And in the end, to combine all this data, learn all this data. And this is on the next slide, big data in agriculture, meaning genome, environment, genome and environment making phenotype. I not make it more complexity, added also management, which is finally farmers see on the field. And again, now is I'm closer to end, and this is my dream. I thinking by 2030 to make potential selection using of genomics tool and keeping diversity, and make selection already on microspore, which is difficult, which is impossible currently because we destroy microspore, but if you could make it undistracted, undestroyed method for DNA extraction, we will be able to improve in some case or applied genomic selection in some case to add gene editing. Important message for me to send you, multiplication and specifically final test, field test should be done on target crop cultivation region. This is what we could not predict by using genomic selection and all other tools. We need to test final product finally on the cultivation condition where the farmers will grow that. And I hope this is again my dream. Please not put it on your notes somewhere very precisely. We could may short the time to 2, 3, or 4 years. And finalize that, I like to give you a few contacts at KWS, myself in a new role, Globally Scientific Affair. Klaus Older, I think very well connected to Barley Hub and our KWS R&D ambassador for James Hutton Institute. Unfortunately, Gabriela was not able to today to join, but we may have next year specific Barley talk. And in this stage, I also like to thank Luke and Joanna for joint supervising PhD students in Bariton project. And before I finish, I put it all 3 books here. Here I was together with Professor Thomas Medaner editor. The book has the same title I mentioned to you. Another book on barley, Johan Kumle and Emil Stein editor. We have chapter on that. And in Raya book, Robbie, I have 3 chapters with my colleague. With that, I like to stop, and I'm really open for your questions. Thank you.
Thank you so much. Just double checking.
I see Alexandra have already hand.
Should we start with there then? Andra, have you a question? If not, I know Pierre Pan has one.
Thank you, Victor, for this very nice overview. Very exciting time, I would say. You talk a lot about diversity and potential use of CRISPR or genome editing in general. We have more and more genome sequencing available. As you know, we talk about the pan-genome coming soon, I hope. Do you think there will be also, let's say, new perspective in terms of exploring or using even more the native traits that probably we underestimate, and to have to really utilize this in a better way than we are doing today? So in addition to CRISPR, I mean, to leverage even more the native traits.
Thank you, Pierre, for this question. I mean, it's related to long discussion we have last week after presentation of Neil Stein on a SHAPE project, and question was how many genome we need for pattern genome to know everything about structural, and we need to have a lot. And I could not, because I'm not directly involved in this work, we see already now many surprise to adding some more genome inside. That's mean I'm my personal view, and I'm very happy to have such large projects like GeneBank 2.0 or 3.0 now SHAPE project, because this has helped us a lot to understand the structural genomics. But structural genomics is only beginning to move into functional genomic. And you remember iceberg I show you, this is upper part what we know on functional genomic. Because I fully agree with you, we still and we will have always a big potential of plant genetic resourcing. I'm personally not believe, and I'm like underline, but this is my personal opinion. This is gene editing is not only the solution, this is additional tool. Was very funny story. A couple of months I have talked with make lecture for very well educated students in plant biology in Belgium, and they're so good in plant biology, and one could have a lot of really nice questions. And one question was, do we need still plant breeding if we are so successful with genome editing? My answer, and I like, and to underline this, is yes, always we need plant breeding, because all tools I mentioned to you, molecular breeding, genomic selection, gene editing, is supporting tool. And this is very dependent of the traits, very dependent of the situation. Classical breeding or modern breeding, we could rename, was, is, and will be. And please remember a slide I show you specifically for farm to fork, because we need to have all possible tools, and plant genetic resources is our goal. We may really use more intensively by using new technologies.
Thank you.
Victor, Robbie's put a long question in the chat. I don't know if Robbie'd rather just ask it himself rather than have me read it out.
Well, it's either you read it out or I read it out. So first of all, great talk, Victor. Thanks very much. So the question was that there's a massive amount of genetic variation in the barley gene pool. But if I understood correctly what you said, that you don't want too much of it in the breeding program. And, presumably that's because of the negative effects that come along with wild germplasm and integrations. And that's probably largely due to the current strategies that are based on integration of genetically simple traits into an elite background. The question really is, though, in the longer term, are you convinced that the current elite background is the best that we can do? And the reason I asked the question is because it's a very, very small and historical selection of the available genetic diversity that's present in the germplasm. It's a by chance selection, right? So are you convinced that there's nothing better out there? That's the first question. You can answer that one if you like now.
Yeah, thank you, Robbie, for this question. Probably to avoid any misinterpretation. What I mean the targeted amount of variation in targeted program. Meaning, I mean, we may start with definition of pre-breeding. Pre-breeding in one public institute like INRAE or JKI in Germany is a little bit different to pre-breeding what is in company pre-breeders done. Because in company pre-breeding is more related to product development already. That means again, to have too much variation is disadvantage for product development, but we should have a sufficient amount of variation for specifically targeted program. Please not misunderstand me. You're fully right. Do the domestication. We may, and on barley, you really nicely shown, and Martin Marshall, some other colleagues show really nicely, we may lose some allele which we need. This is mean excellent opportunity for us coming now back by using all techniques we have on genomics to try to recover that. In two different ways, integration from plant genetics resources, and barley would be mean second or third genetic pool, or applied gene or genome editing for the allelic variation we may create for specific targeted gene. But no any doubt, variation is crucial for any breeding program.
What do you think about the idea for having a completely de novo domesticated gene pool based on the wild germplasm?
I don't know. Somebody should try first.
Okay. Good answer. I'll let other people ask their questions now, Luke.
Just checking anyone else got a hand up. Or I was going to ask a question, Viktor. Given your experience across the cereals, is there something that the barley community could learn from your experience in rye, looking at an outbreeding crop?
Luke, thank you very much for this question. I am really like, because I'm completely forgot to make the promotion for rye. Now, to be seriously, if you look now farm to fork and condition we have sustainability, use of nitrogen, to be more resilient to abiotic stress, rye always on such target is very important. I mean, nobody from scientific society make for me still answer why rye could survive by -35. We are talking about the same cluster of the gene on chromosome 5H of barley or 5R in rye or chromosome 5A in wheat. We know the different orientation, but nobody from scientific society could explain me why rye could grow by -35 and wheat probably by -25, which is very famous North Star from Canada. But if you know why, we may have the potential to shift it winter wheat in Canada much more, which is mean more yield. And for barley specifically, because in some case, Polish farmers have problem with winter barley because winter hardiness is not sufficient enough. Drought. Rye is more resilient to drought. Why? Root system. Rye is much more have deeper roots and use more efficient nitrogen. Question again, why? That's the reason I still see a lot of potential we need to learn from one crops to other. And in Dundee, you have unique possibility. You are so well developed on barley, you may use some study on rye as well. And one last thing about, if we are talking about hybrid barley or hybrid wheat, we need to learn from rye, because from biology of rye, just give you one example. Pollen fly on rye 7 kilometers, for example. In wheat, it's not fly at all, it's too heavy. And the question is, what's the way of biological? Yes, we have some variation, but is sufficient this variation, or we need to have probably new creative idea from scientists how to learn wheat pollen to fly a little bit. And I think this is win-win situation from one to another crop. Of course, on barley is much easier to work because self-pollinated crop. You have excellent tilling population. We tried on rye to 3 times, at least on my knowledge, to make tilling population is not successful, because I think evolution of rye speak against to put it immediately in homozygous stage. But definitely look, once again, I think we need to learn from one crops to improve another crops as well.
Yeah, thanks. There's another question from Robbie. Anyone else want to chip in at the stage? If not, Robbie, you have another question?
I could keep asking questions if you want, but what I was going to ask you, Victor, was that how many killer genes or traits do KWS have lined up in the pipeline that you'd like to modify using genome editing? And do you think any of these traits would be sufficiently important to convince the public that genome editing is the way forward?
Thank you, Robbie. Again, very important point. One sentence before I will talk about the traits. I think it's our responsibility as industry, but also your responsibility as scientists to bring specifically in Europe product ways not only farmers but consumers will be mentioned. Finally, scientists did for us something positive. I'm talking because we have so deep, how is called English word, against of GM, and because we have enough food in Europe, we not care probably too much about that. And this is very important before. I believe personally the technology have potential and big potential, specifically if we are talking about sustainable and resilient agriculture. But this is not replacement of plant breeding. This is a tools which is by using these tools, we may be more quickly, more precisely, and finally more successful with product to consumers. To answer your question about the traits, I would not disseminate or not explain in all details what kind of traits. Yes, we made decision. This is very crop specific, because for crop like corn, maize, we have different targets as for wheat or barley, and we are very actively working on that. And for us was the decision of European Court was not decision for us, because as you know, we have second research center in United States. We continue with all genome editing related research for most important our crops, including rye, for example. But traits is different to the crop. And again, I like underline the second reason why not explaining about the traits, because again, the motivation I mentioned for you in beginning is our joint responsibility to bring on the shelf of consumers some product which is farmers and finally end consumers will be mentioned. This is excellent. We are really loved. I mean, we could spend a lot of time on discussion about that, because acceptance on red biotechnology taken of vaccination as last example is completely different. And that's the reason is why we need to have really important case of, and we know case is also in public domain, you know, in UK, you know, in France, you know, in Italy, for example. The biggest project just finished on gene editing.
Okay.
Not going on the traits discussion.
No, it's just that from all of the bad press that GM had, I just wondered if breeding companies per se had a series of traits that they thought the general public would really embrace as, you know, being something that was not achievable using standard breeding technologies, because I think that's going to be the proof of the pudding, actually, of whether it's actually accepted in Europe. You need something which is going to be really good.
To make it short joke,
benefit.
To make a short joke about this, this will be definitely not for cereals herbicide resistance.
Yeah. It always astounds me why people are quite willing to have some genetically manipulated DNA or RNA injected into their arm, but they won't eat anything that's got a small amount of genetically manipulated DNA in its flesh that you eat. So yeah, it's just that it's curious. It's curious.
But take it back to the question you asked in beginning to use a secondary pool of barley, for example, to introduce. This is also the possibility potentially which could be done by genome editing. Yeah. If it knows the function, if it knows allelic variation, we may create for specific targeted traits additional allelic variation. Yeah. To make the short way of introgression.
We also have a representative on the call here who have got a technology which might be a rival for genome editing anyway. So I think it's important that we keep our options open for how we want to achieve some of the things that we want to achieve.
Absolutely.
He may want to say something, but he may not. So I'm not even going to point to who it is.
Maybe else for some questions? Ah, yes.
Yeah.
Uru.
Yeah. Hi, Victor. Thanks for the interesting talk. Well, I just want to hear your personal experience on serial breeding in the era of genome sequence, because you've been working on the past 2 decades, and the reference genomes are available in recent times. Yeah. And what kind of changes or innovations that genome sequence really brought into the breeding program? Not just genomic selection or changing.
Yeah, thank you. Is a difficult question to answer. Short answer will be molecular breeding is part of breeding process, all our breeding program on zeros. Second answer, yes, definitely sequencing of genome help us a lot on development of allele specific marker and so on to make the molecular breeding is extremely precise link. But still talking about barley genome or on rye, we have a lot of repetitive DNA, which is 30, 50 years ago, we mentioned this is rubbish, it's nonsense. But now we are recognized more and more, and I came again back from annual meeting of funded PMBF project, and one big call we have on epigenetics, which is still a very difficulty in breeding specifically to understand and we don't have the tools how we could measure that. But I think is still epigenetics also could pay additional advantage for plant breeding, specifically on the plant environment interaction, specifically how plant adapted to one or another condition. Yes, no any doubt that without sequencing we will be not successful as we successful yet for different dimension for current modern plant breeding program and zeros, but also potential for genome editing. But also we still have a lot of to do, and I already underlined how important will be functional genome. And again, remember you about amount of funding going to human genome research. And human, as Homo sapiens, we have only one species. In plant, we have many different species. We have also competitions on funding, on resources for one on another species. And this is we should be very smart enough how we combine knowledge from one species, like Robbie asked, or Luca asked about what we could learn for rye or opposite way around, to be smart enough to use this knowledge finally in form of translation of knowledge for practical plant breeding. And for that, and I forget to mention in my talk, I'm very thankful and I see a lot of potential, because in Germany I was as pioneer in started of that on public-private partnership. Because for us as KWS is also very crucial, and we are very happy to work with our competitors, also with our friend on pre-competition research, which is make win-win situation instead of spending money twice or 3 times to work together to see what we could do on domestication, like Robbie, you mentioned, to try to introduce from secondary pool to elite pool now again, some allele which we are probably during domestication forget it. And this is exactly what is our strength in Europe. And I believe we could do more.
Thanks.
I think with those words, I think that's a really good way to finalize this talk. Thank you very much, Victor, for your enthusiasm as always, and thanks for the talk. Just want a quick word from Robbie there, a hand up.
But before Robbie mentions the final word, I like also to thank everyone who was today. I was so happy to see a lot of my old colleagues. Of course, I would prefer to see it face to face, but I hope still time will come. Thank you very much for your attention. Thank you very much for your interest. Robbie.
My hand up was actually a mistake. I was trying to get the hands to clap for you. But let's just finish by reminding people that the next talk for our community initiative is exploring promising candidate genes for barley drought tolerance. using functional and translational genomics, and that's by Agata Daskovska from the University of Silesia. That's on the 13th of April at the same time. So if you're very, very welcome that everybody just to come along and join in again. Been a great talk, nice session, Victor, well done. And yeah, pass my regards to all of our colleagues in Einbeck.
I will do. Thank you.
And great to see so many old friends and faces. So lovely to see you.
Thank you very much. Have a nice afternoon. Bye bye.
Take care.